All right, welcome to the Church of Perpetual Life. So glad to have you here. I know more people are coming up, but it's time to get this event going. And my name is Neil Vandry. I'm your officiator here at Perpetual Life, and I'd like to welcome you as well as everyone who's online. We have a lot of people who are joining us in the live stream tonight. Here at Perpetual Life, we refer to ourselves as immortalists, not because we have conquered death at this moment, but we have faith in humanity to do so in the near future. I'd like to show you the Perpetual Life Creed, and if you'd like to read along with me, you're welcome to. Here at Perpetual Life, we believe that all of life is sacred and that we have been given this one life to live, to make unlimited. We believe in our Creator's divine plan for all of humanity to have infinite lifespans and perfect health and eternal joy, rendering death to be optional. By following our Gospels, we achieve eternal life, creating a heaven here on earth. We follow Nikolai Fedorov, who taught that the transcendence of the Creator will only be solved when humanity, in our unified efforts, become an instrument of universal resuscitation, when the divine word becomes our divine action. And we follow Arthur C. Clarke, who said the only way to discover the limits of the possible are to go beyond them into the impossible. And so we enter each day energized in spirit and empowered by the words of our prophets to live in joy, serving our Creator and all of mankind forever and ever. As you know, the RadFest was in October, and they have now got online their video presentations. So if you'd like to see the video from the RadFest, just go to radfest.com and you can get those videos. Our next service is next month. We have typically one service a month, and that is February the 27th. We have a presentation by John Asher titled The 9 Steps to an Unlimited Life. So be sure to come back on February 27th. I'd like to ask by a show of hands, who's here for the very first time tonight? For the very first time, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, that's great, 13. I want to welcome all of you specifically, and I hope that we get to see more of you. Some of you came as far away as Tampa. What a drive that was. So glad that you are here. Anybody come farther than Tampa? You are tonight's winner for distance. All right. I want to point out also we have a lending library right outside the outer sanctuary. If you'd like to check out a book and bring it back next month, next time you come, be sure to look at that library. Downstairs we have some t-shirts and books for sale by a donation of $8. And there's also a table here as well as downstairs where you can find information on cryonics. If you're curious about cryonics, we have information here at this table as well as downstairs, and feel free to talk with me after the event tonight. So now I'd like to bring up our first speaker because we've got 2 speakers tonight, and we're going to have a great night. Liz Parrish is the founder and CEO of BioViva Sciences USA. BioViva is committed to extending healthy lifespans using gene therapy. Liz is known as the woman who wants to genetically engineer you. She is a humanitarian, entrepreneur, author, and innovator, and a leading voice for genetic cures. As a strong proponent of progress and education for the advancement of gene therapy, she serves as a motivational speaker to the public at large for BioViva and the life sciences. She goes all over the world giving talks. She's actively involved in international education media outreach and is a founding member of the International Longevity Alliance and founder of the BioTrove podcasts found at iTunes. Let's welcome our speaker tonight, Liz Parrish.
Can you hear me? Hi, thank you so much. I feel so sorry for people when they have to say all of that. There's so much room for error. All I want you to remember is that I'm the CEO of BioViva, and I do want to genetically engineer you. It's true. And I'm going to talk about why I would want to do that, because that sounds like a very invasive thing, but actually it's a pretty simplistic process, and it's the way that we will ensure that we live a long time in good health. So life is a timeline, or a cell is a timeline. Cells have a limited capacity of life, but they actually build us. We have trillions of cells in our body that we rely upon, and we rely upon them staying as youthful as possible. So when cells get old, bad things happen. And today that we know that not only humans, but a myriad of species have what's called the Hayflick limit, meaning that their cells can divide a limited number of times. So that limits our lifespan, and it limits the lifespan of a bunch of other creatures as well. So a bowhead whale might live to 250 years, but a human might live at the maximum up to 120 in the best health. But my life is also a timeline, as is yours. We all have a story. And I'm going to tell you a little bit about my story and how I got here and got so lucky to speak at the Church of Perpetual Life. That is so cool. So in 2011 to 2013, I was asked to volunteer my time to educate the world about the use of stem cells. And this came at a really advantageous time, as my daughter had just come home from school, a very excellent school system, and told me that they had taken a poll in class about stem cells. And she said, over 80% of people in my class think it's unethical to use stem cells. Whew, really? Well, that's because people at that time, and some people still do, think that stem cells are fetuses put into a blender, and that's not true. So I joined a nonprofit, and for 2 years educated the world about the use of stem cells, and that most of the stem cell use, over 95% of it, is autologous stem cells. They're stem cells from your own body, used in your own body at that time, and that embryonic stem cells were in fact not fetuses. Why did I stop doing that, and what happened? Well, one thing, I fell in love with the stem cell. I fell in love with its genes. So every cell in our body has the same genes in it. But we have noses, and we have toeses, and we have spleens, and we have livers. So how does that actually work? Well, it's called epigenetics. Our genes code differently in different cells of our body, and I fell in love with the genes in stem cells. Why was a stem cell able to regenerate tissue? Why could it turn into any tissue in your body? And yet other cells in your body, when they were damaged, were just done. Or worse yet, if they got cancer, they just went on infinitely dividing until they killed the host. Well, it was these epigenetics. So I started meeting with epigeneticists, and they told me about all these fascinating genes, the differences between cells. And I accidentally fell in love with genes. And now my love affair continues because these genes, the manipulation of genes, can make cells do amazing things. In 2013, my advocacy for the use of stem cells comes to a crashing halt. My son is diagnosed with type 1 diabetes. This is actually him in the hospital. And I looked around and I said, okay, we're going to advocate. We're going to get this new technology that I've spent 2 years learning about. I've seen all of this research and development. I've seen what this technology can do. They said, write on a board your three questions that you'd like to ask your doctors. And my questions were all based in what can you do with stem cells? What can you do with genetics? And can you biopreserve his pancreas? So they came in, and one after another, we saw many doctors. When you are diagnosed with type 1 diabetes, you're admitted to the hospital, and you have to live there for several days. You've got to live there for about a week because you have to relearn to manage your life. It's not like type 2 diabetes. There's no diet and exercise that can help it. It's an autoimmune disorder in which the immune system attacks the pancreas and destroys it. You're immediately insulin dependent. So they looked at me and they said, whoa, lady, that stuff is experimental medicine. There are kids here that are dying. Your son has a manageable disease. Be happy. It never fit into my head that there were kids there that were dying. That we have all of this technology that's been in research and development literally for decades, and we don't use it in humans, and they lose their biggest asset because of risk aversion, because we're afraid to use the technology. So I got on an airplane and I started to go to any genetic conference that I could find. I wanted to find cures for kids. I did not initially get into this to cure aging. I didn't know anything about it. I found out that treating aging was a miraculous thing. I found out that even when we're 80, we feel like children and we don't want to lose our biggest asset. And every gene therapy that my company looks at can also treat a childhood disease, because aging, the cellular degeneration, and other diseases of childhood go hand in hand. Probably the most succinct connection between the two is a disease called progeria, which is accelerated aging in children. And by lengthening telomeres and a few other the therapies we're looking at, that might actually cure that disease. So I find this connection between aging and childhood disease, and I learned an interesting fact that over 100,000 people die every day of biological aging. That's 100,000 people that might have been offered technology that could then help everyone live a healthier life, help children see their first, and help them see many more firsts, things that we haven't done yet in our lives, things that we haven't explored. So we're going to jump forward a couple years, and in 2015, after 2 years of studying, from 2013 to 2015, we looked at, my company, BioViva, was formed. We looked at the 2 most promising gene therapies to treat biological aging, and it's in September of that year I took those therapies. And that's me going, when is this ever going to end? I was injected with telomerase inducing gene therapy, and I was also injected with follistatin gene therapy. And we're going to talk about those gene therapies later in this talk. But I was so excited to maybe revolutionize medicine, to show the world that we were sitting on the greatest technology. It was not being used, and we might be able to save 7 billion lives. But we're going to go back and talk about what is aging, because I want you to understand what we're treating to begin with, and I want you to understand why I think aging is a disease. As a matter of fact, I think it's the most succinct pathology on the planet. This is actually fast forward. In the 1600s, the first death records showed us that over 80% of the population died of infectious disease. Those were the first death records that were out there. Some people, about 1% of the population, died of something called aging. And there were many poets at the time who wrote about this other type of disease, this sort of just lasting until you couldn't exist anymore. But in 1900 in the US, this is what the death poll looked like. 53% of the population still died of infectious disease, even though we had gotten a pretty good handle on it at this time. But what does science do? Science moves forward. Science tries to solve problems. And by 2010, only 3% of the population now dies of infectious disease in this country. This is science at work. So every time you get an immunization or an antibiotic, if people would have told you, if you would have had to sign a waiver that said you are now artificially extending your lifespan, we would think a lot differently about science and the technology that we take. This made a huge impact on how long we live as a population. And if we want to look at aging itself, we're going to watch these lines appear. When do we get the diseases that we predominantly die of? We generally get them over the age of 45. By the year 2050, aging is the biggest killer on the entire planet. That's in every single nation. So here you're seeing your risk, and you're seeing something even more interesting that I'm going to tie in later. Today, actually it's 2020, guys, congratulations. And this year is the first year in the history of humans that we have more people over the age of 65 than under the age of 5 on the entire planet. except for the 5-year-olds are vastly not being born in many of the countries. They're being born in diverse countries around the world. They go on to be the healthcare payers. So the 5-year-olds become 10 and 15 and 20, and we become 65, 70, and 75 and 80. We don't have enough payers for this system. And we have some really interesting documents if you want to look into some of the things the company is doing about the GDPR and the actual cost of aging just to the United States alone. It's chronic. It's something that we can't afford. All of the diseases of aging are now considered a $1 trillion disease, some of them globally and some of them independently in each country. The cost is really high, and so developing technology that people can take, even if the technology started out very expensive, would be cost effective in the long run. But what is aging itself? This is a really neat little video of slides. These are 4 sisters that are in Oregon State, and every year they get together and they take a picture together. And essentially for us, what it shows is their great beauty, and their great beauty over time. They're still as beautiful today as they were before, but it shows what happens about how we think about aging. So when you and I think about aging, we think of external properties of aging. We think of wrinkles and fine lines and atrophy of muscles. But aging is more than that. These sisters actually do still continue to take these photos, so mine are a bit outdated. But isn't that beautiful? That's life as a timeline. And these people have accumulated information like you've accumulated a multitude of information over your life, but when we die, we lose all of that. I think of it as like being the worst book burning on the planet. Unfortunately, aging is also happening on the inside of our body. So this is the brain of a 27-year-old and an 87-year-old. And this 87-year-old does not have dementia. This is just what happens to the brain. And if we looked at every organ in the body, from the liver to the heart to every organ, we would see these type of changes over time. Aging is at the center of this, and the geroscience hypothesis is aging is the biggest risk factor for mortality. So what are we going to do about it? We can do things about it, and there is evidence that we can do amazing things about it. My company looks at the hallmarks of aging. So these are things that are happening at the cellular level. They're happening both inside the cell and on the outside of the cell. And they are the reasons that we age. So remember, we're all for chronological aging. We want you to get older by years, but biological aging is a detrimental process that is very harmful to the organism. So here we'll see things like senescent cells, and you've probably heard a lot about those lately, especially in Life Extension magazine. Bill does a great job of keeping everyone up to date on the newest research. Genomic instability, telomere attrition. Your telomeres are getting shorter with every cell division, and they are what's linked to the Hayflick limit. As your telomeres get short, eventually your cells can't divide, and that's generally the end of the organism. Epigenetic alterations, the epigenome that we talked about changes, loss of proteostasis, deregulated nutrient sensing, and mitochondrial dysfunction, which is a hallmark of every one of the diseases of aging. If we look at cancer and we look at which hallmarks are affected in cancer, these are the ones that are affected. And this is really important. So we are going to walk through 4 diseases right now and how they are mapped onto the hallmarks of aging that are affecting you. Dementia and type 2 diabetes, they share the same hallmarks of aging. There was just a paper that came out just a couple days ago that talked about this, the link between dementia and type 2 diabetes. It's a cellular link. And cardiovascular disease, one of the biggest killers on the planet. This is a Venn diagram of those same 4 diseases. And you can see why treating biological aging is the smarter step to take, because these 4 diseases alone kill over 80% of the population. But what about the battle against cancer and the battle against heart disease and a myriad of other, nonprofits that are out there? It's all fantastic because it shows that we all agree that we want to and we desire to cure these diseases. They're inhumane, they cause a lot of suffering. But if we had an 82-year-old woman and today we cured cancer, which is a great thing to do, please don't stop doing that. It's a great thing to do. We would only add a few years to her life. That's not expected, was it? If we cured heart disease, we add about 4 years to her life. And if we did a combination, we could add maybe up to 16 years, but if we just slowed aging, we could add over 33 years to her life. And that's because the hallmarks of aging that we just saw are all happening in your cells. So people over 65 usually have 2 or more diseases of aging, but you're just getting treatment for one of them. So what I'm trying to tell you is these are all succinctly correlated. They're not even independent. It's biological aging that is causing the symptom of the disease that affects you. So this is old therapeutic intervention. We used to look at, and we still do, unfortunately, look at one therapeutic to target each one of these diseases. But if we target aging, we'll get the biggest bang for the buck, we'll have the biggest outcome, and we can extend healthy human lifespan. How do we do this at BioViva? So we look at gene therapy. This video is showing you an AAV. It's a vector. It's a gene therapy delivery method. And what we do is we have it deliver a therapeutic gene that upregulates a protein that changes you. So remember, all the cells in your body have genes. They all code for protein. They're why you look the way you do. They're vastly responsible for a lot more things than we thought that they were responsible for in the past, even personality traits. And if we upregulate genes that have been around for decades in research that regenerate the cell, that make organisms more youthful and healthy and viable for a longer period of time, it radically changes how these cells, these human cells, behave. AAV gene therapy has been in over 400 trials now, and so thousands of people have taken gene therapies for a variety of generally monogenic disease, meaning one gene defect. If you're born with a one gene defect, your chances of having a cure through gene therapy are actually quite high, and there's 5 regulated past gene therapies that are just that. They're a one-time treatment for a lifetime cure. 5 of them. Boy in the bubble disease has been cured. Spinal muscular atrophy has been cured. These children are taking 1 injection. That's so fantastic. That's the power of the technology. But the technology is vastly expensive, and what do we want to solve? We want to solve the rate of translation, because it doesn't really help us very much today. No, it doesn't help anyone in this room if you don't have a monogenic disease. So drug discovery today is very slow, it's cost prohibitive, and it's very likely that you won't succeed even if you have an excellent drug. It's called knowing your endpoints. So if you don't have human data and you have a bunch of mouse data and then you go into clinical trials, today the success rate is dismal. You have a 94% failure rate in phase 3 alone. But what if we could turn that on its head? What if we could find the endpoints of therapeutics in humans, because humans today need access to this technology, and we knew our endpoints going into clinical trials? That is the goal of this company. Number 1 is patient access. We want patients to have access to the technology that they need today. Number 2 is data science, getting that data into a database with bioinformaticians that can sort out where these drugs are working and where they need to actually be targeted for clinical trials. And then the last one is research and development. We work with Rutgers University, and we're developing a new delivery method for gene therapy so that we can get multiple genes in predictably at one time. So again, these are today's gene therapies. These are the regulated gene therapies. They have been passed for use in humans. And one thing you might want to note right away is the cheapest one is $425,000 for one eye. One eye. That $425,000 means essentially you could buy a nice house or you could see out of one eye. and this is a problem. And how you solve this problem is you actually treat the biggest unmet need on the planet, which is biological aging, and you do it by scale. So when you have one patient that has a rare disease, you basically have a very expensive therapeutic, especially if it's gone through regulations, because all the shareholders want their money back for the development of that drug. But when you treat the biggest unmet need on the planet, meaning everyone in this room could benefit from the gene therapies I'm going to tell you about, we can get the cost down significantly. And how we're going to do that is by using Integrative Health Systems. So Integrative Health Systems is an exclusive partner to BioViva, and they offer gene therapies as medical tourism today. So they offer the ability for people to access technology that you couldn't get anywhere else in the world and get it into humans who need it today. And what's really fantastic about this is nonprofits are stepping forward to help us. Today we have a trial for dementia that for 10 patients is 100% paid for. And these are really, really expensive technologies. That's each one of those patients gets about $50,000 in gene therapy. The conditions that they're looking at treating today, again, is we have it listed as Alzheimer's, but actually it's a dementia trial. We do muscle regeneration for people who are frail and aging because people die from falls. My best friend's mom just recently died from a fall in June. We do Clotho for kidney regeneration. It's also for cardiovascular disease, and it is known to increase intelligence. So you wouldn't be surprised that we get contacted by a bunch of smarty pants who want to be a little bit smarter. Darn them. And then we do telomerase, of course, which is one of the gene therapies that I took, and we do it for anti-aging purposes. And we have dual and tri-gene therapies and all sorts of fantastic things. But really quick, we're going to talk about the genes. The first therapy we're going to talk about is the telomerase induction. So telomerase induction has predictably reversed aging in human cells over and over. It's extended the lifespan of model organisms. It affects almost all of the diseases of aging positively. You're conceived with about 15,000 base pairs. You're born with 10,000, and by the time you're diagnosed with your end-stage disease, you have about 5,000 base pairs left. So a lot of people ask me, does telomerase induction cause cancer? We always go to our advisors on that. Bill Andrews was our advisor for years, and he says no. And the research shows that it does not. And I have it all throughout my body for almost 5 years now, and I have no increased cancer risk in any of my testing. We think it might actually protect against cancer. The second gene therapy that I took, and I really love, because this is one of the lowest hanging fruits. It's been through safety and efficacy with other companies in the United States. It was used for Duchenne's and Becker's muscular dystrophy. It's called follistatin, and it increases your muscle mass. As a matter of fact, statistically through clinical trials, 8 out of 10 people benefit from it. We're not sure why the 2 odd players out don't. It increases the muscle mass, and that is a great defense against type 2 diabetes. Increased muscle mass means better insulin sensitivity, and we have shown that over and over in patients. This boy on the left has muscular dystrophy. This is the same boy on the right 3 months after treatment. Yeah. So, I mean, that's really fantastic. And I believe that this trial was done by Pfizer, so I'm not going to claim ownership of this. This is not about ownership. This is about sharing data and helping people. And that's the way we hope the world works moving forward. Our third candidate is called Clotho, and it's named after the goddess Clotho, who spun the web of life. The gene actually codes for the protein, and the protein was found to be deficient in people who had chronic kidney disease. On closer inspection, it just so happens that people who have this naturally upregulated in their body are not only protected against chronic kidney disease, but they're protected against cardiovascular disease, and a new paper out in August showed that they're also protected against dementia, even if when they die and after autopsy, it's shown that they had full-blown Alzheimer's. So in our dementia trial, we're doing 2 sets, one set that gets telomerase induction, the other set that gets telomerase and Clotho induction, to see if we can have an effect against the dementia decline and cognitive enhancement through this gene therapy. All of these genes extend life significantly in animal models. And Clotho upregulates something called FGF21, which one of our scientific advisors recently, a group he worked with, came out with a paper to show that it extended lifespan significantly in animal models. That's super fantastic, and that was reproducible. So one thing I want to make really clear is we don't just pull these off of the shelf and they've been in one animal study and we think this is great. We actually do what's called reproducible science. So we make sure that more than one laboratory has done work with the gene and they have all had a predictable result coming out of it. So then it's not so biased and difficult to understand. This year, late in the year, PGC1 alpha goes online. PGC1 alpha helps with mitochondrial function. It makes your mitochondria more robust. Your mitochondria are the powerhouse of your cells. Think of them as little batteries. They make you run. And this gene therapy has been revolutionary. It's increased lifespan in animals, and it actually increased energy production. So when you put these in little mice that are obese, they become the correct weight, and they run much farther and longer than their peers. Remember, they're on a wheel in the lab, and yet they live significantly longer. And so this is a really exciting gene. And we've got a couple more, but we're not going to talk about them because it gets kind of overwhelming over time. So gene therapy, right now, what we do is we help people get access to this technology who need it now, but how will this be given in the future? It will be given like an immunization. You'll get these gene therapies long before you suffer any aging effects in the future, and I'm sure about that. As long as we stay on the right route, that's just where we're going to go with this, because you get the biggest bang from the buck from not getting sick to begin with. Right now, we have a sick care system, meaning we wait for you to get sick, and then we treat a symptom of a symptom. So we treat beta amyloid plaques, or we treat, atherosclerotic plaques, which are a symptom in the first case of Alzheimer's, and the second case of cardiovascular disease, which are just symptoms of aging cells. So they're just going to keep coming. So step 2, data science. So it was supposed to go live in November, so I'm here to say that we are definitely suffering the effects of being a new company, but it'll hopefully go live next month. We are putting up a multi-omics repository that people can come to, and even if they don't want to participate in gene therapies, you can store all your health data in one place, and if you allow us to, you have to check it off, we can use your data to help cure aging. So it's a way that everyone can get involved in helping us look at what biological aging is. And what's really cool is you get to be the people who are the controls for our gene therapy patients. We want to see if the people of the same age as you, how they're performing on gene therapies. So that makes it so that anyone who comes through our platform actually gets a gene therapy, and the controls are outside the platform. You can store a myriad of data, your telomere data, your genome, your microbiome, your health data records from your doctors. Again, you don't have to share it with us, so we're doing the value added of you can just have all of your medical data in one place, so you don't have to log into 5 different websites to get it. And then this is what our researchers will see on our side. They'll be able to analyze your data. They have a lot of platforms that they can put your analysis through in order to see how you're performing and give you some valuable feedback over time. It's HIPAA and GDPR compliant, and we think that that's fantastic. And then we also are offering a DNA methylation kit. It's actually officially launched, but our website's not up with the BioVault yet. And it essentially tells you your biological age. So methylation kits are your most accurate biomarker for biological aging. So I have a friend in the audience, Robert, and his family lives to be very old, and they're very healthy for a very long time. So biologically, they're running probably a lot younger cellularly than the rest of the population. But someone at the same age as Robert or his mother may have a completely different profile and be in imminent danger. The great thing about your methylation kit is you can affect it. So there's evidence that therapies can affect it, and there's evidence that lifestyle can affect it as well. So you're not stuck with this data, so it's kind of a good thing to know and then learn how to actually massage it back a little bit. Okay, and then lastly, we're going to get into research and development. So with Rutgers University, we have been working on a new viral delivery method. Yes, they're viruses, but they can't get you sick. We take out their ability to get you sick and we put genes into them, just like the AAV vector that I showed you earlier, that can then upregulate good proteins. And this just goes to show that, this is the reason that we would do any animal studies in our company is because we have to do basic research. So we have to show toxicity data in animals. Even though our vector has been in humans for different reasons, we have to start all over. And we choose mice, not because they're a great model for humans, but because they don't live very long, and we can see how long that they will live when we give them certain interventions. Our gene therapy is quite amazing because it's quite large. With AAV, you'd have to do, if you wanted 3 gene therapies, you'd have to do 3 different therapeutics. You can do them at the same time, but we don't know what cells get which genes. With this one, we can ensure that all of the cells get the 3 genes that we're actually targeting. And it can be delivered in multiple ways, and it doesn't give you an immune reaction, which is, or it's negligible anyway, and that's what a good gene therapy does. Our lucky mice. So when our mice were treated, they were actually 18 months old. So the human equivalent to that is between 58 and 68. So that's really, a good target. For human studies, they were wild-type mice, so they have that nice wild-type genome, and they were all females. There were 6 cohorts. We're working on a proprietary way to deliver gene therapy for neurogenic disease, so some of the mice got that, some of them just got the gene therapy systemically, and then the ones who didn't get it are almost all dead now. It's been months into this. And I can't wait. You may not care, but later this year, or depending on how long these mice live, in early 2021, we'll be coming out with the paper. And when you see them, you will not believe it. They are lovely. They are youthful and fantastic looking, and I'm just gushing over our mice, which is kind of funny. This is the distribution of the gene therapy. This is our scientific advisory board. So we were lucky enough to, right at the get-go, upon the inception of the company, knowing that what we were going to do, get, I think, the best scientific advisory board on the planet for aging. I don't have one of our really big advisors on here, but this over here on the left, if you don't know him, you should. That's George Church. He's the professor of genetics at Harvard University. He's well respected in the area. He's been looking to treat aging for a long time, and he helps with our Rutgers project. And then we have Aubrey de Grey here, which I think he's come here to speak to you, and he's kind of like the master of all things anti-aging. It was actually his conference that turned me on to treating aging instead of going straight at childhood diseases. And then we have a myriad of doctors and scientists, and on the right we have Adam Sandberg, who is at the Futures of Humanity Humanity Institute at Oxford University, and they help us de-risk ideas, make sure that our ideas actually will be fruitful and turn into a better outcome than a negative outcome. They literally look at everything from asteroids to, pea gravel falling in the wrong place, a butterfly wing. So that's really fantastic. So these are the three groups that we work with. Of course, we're BioViva on the right. We work with Rutgers and Integrative Health Systems. But really quick, before I am done, I wanted to go over some of my data. This is an older presentation, so I'm not sure how up-to-date it is, but I know that it has some of the answers to some of the questions that you might have. So I took telomerase induction and a myostatin inhibitor. So again, telomeres are the caps at the ends of your chromosomes, and they get shorter with every cell division. And that division has a limit. Telomerase is the enzyme that lengthens the telomeres on the caps of those chromosomes. Telomerase is turned on in your germline, keeping you having youthful, healthy children, but it gets turned off in almost all the rest of the cells in your body, although stem cells have some small amount of it turned on. And on the right side, myostatin inhibitor. So you can come and feel my muscles afterwards if you want. I don't work out, but I have more muscle mass than when I did, significantly more. Trying to look dainty here. And that's a really great feeling. So I was 44 when I took the gene therapies, and people always say, oh, this doesn't have my last telomere data on it. They always say, what did you feel? What was the difference? And it's really the lack of things that I feel rather than the things that I feel now. It's the lack of aches and pains. It's the ability to carry all the groceries in when the kids don't want to help me. I'm climbing up on things. So I would say that, being healthy is really the lack of things that are uncomfortable. So we don't have my last telomere results on here because they lengthened again after this to 8.61 or something like that, base pairs. And it was about equivalent to 33 years in increase. We did SpectraCell all the way through the testing, and then we did some life length tests just for your edification to make sure that the SpectraCell tests were having the right readout, and they did match. These are my muscles before and after. So my before ones, I ran about 5 days a week. I did all of my meetings. Anyone who knew me, I did all my meetings while I was running. And afterwards, I went from one plane to another plane talking to regulators. I got to meet a couple presidents. I got to meet the Dalai Lama. I got to meet a lot of people to talk to them about what we were doing and why we thought it was really important. And I wasn't exercising, but we still saw increase in muscle mass and decrease in white fat, so that was good. My C-reactive proteins, for some period of time, they're still in an excellent level, but they had a 6-fold reduction, and so that meant the inflammatory markers in my body were super low, and inflammation definitely drives aging. It's a symptom of aging cells. And my cancer risk is low. This actually continues to be true in all of the patients that come through. We'll have that data out probably later this year. We see across the board a reduction in blood glucose levels, which is what we would expect with increasing muscle mass, and the A1Cs are really excellent, and a reduction in triglyceride levels, which means better heart health. No liver damage. We didn't get damage in any of these areas. So this was just a slide, stating the outcomes. We didn't have a negative outcome. With gene therapy, so when you take a small molecule, you're taking a pill, it's going to go through your liver, it's going to affect your kidneys, it's going to have all of these what's called side effects, and you're hoping that some of the side effects are on-target effects. They're things that you want to have happen. When you upregulate a gene therapy, you're literally using your cell as a drug factory to just create the target protein that you're looking for. So we didn't foresee seeing negative side effects, and we so far haven't. Cross our fingers. And, lastly, I just want to leave you with, before you ask questions, we really do have to decide what kind of a world we want to live in. This comes down to you and me. There are no heroes. Nobody's going to come and save us. I took this upon myself because my child was sick. I learned a whole bunch of things that I didn't know because I had to. I participated in something by choice because I thought it was one of the greatest things that I could do to improve life on Earth. This precious, precious, special time we have. So we have to decide if we want to live in a world where we continue to tolerate the diseases of aging, or a world in which we step up and we pioneer the technology that will radically change our lives and the lives of the people that we care about. And that's it.
Thank you, Liz. We have a few minutes for questions.
Sure.
If there are any questions in the crowd. And also, I'd like to point out that Liz's PowerPoint will be available on our website in a few days. You'll be able to access it there. So let's go right here to the front.
How is your son?
Well, does anyone in this room know about type 1 diabetes? Yeah, some people know. So they know that it's a disease that you deal with essentially on an hourly basis, so it's common almost every night that we have a critical low, and every day that we battle high blood sugars. He's 16 years old, so he's in a real swing and brittle time, and it's difficult to deal with. And so, I have a whole folder on therapeutics and gene therapies that are coming to affect autoimmune disorders. Some of the hallmarks of autoimmune disorders are very similar. So the type of treatments that might help something like type 1 diabetes might help rheumatoid arthritis, MS, Crohn's disease, and the list goes on and on, lupus. So, the only good thing about watching his struggle and being part of it is that it reminds me every single day to work as hard as I can. Every single day. When I get stressed out, I open the computer. When I get stressed out, I make a call and try to find somebody who will help us. So, the good part of it is that, and he just got his driver's license. So he's on a soccer team. He's been on soccer teams since he was in the first grade. So, things are going normally in his life, except for type 1 diabetes it's an hour-by-hour disease. Every time he eats, he has to take insulin. He has a pump, he's got a CGM injected here, a pump injected into his stomach. I look forward to the day when he's free of those.
Did he get stem cells?
He has not gotten stem cells. He is not really, we've asked him several times if he is, and the problem with stem cells is because it's not a curative effect in type 1 diabetes. It puts you back into a super brittle zone. So if the stem cells start behaving like beta cells and creating insulin, we could have a catastrophic low. So we don't want to be on any roller coaster. So insulin is the sixth most expensive liquid in the United States. It's massively expensive, and it's super deadly. So when you have type 1 diabetes, you create no insulin. And so you have to inject it for the amount of food you eat. And in the beginning of type 1 diabetes, you're in what's called a honeymoon zone, meaning that you still create some amount of insulin sporadically on and off. And it's a very, very scary time. So for type 1 diabetes, until there's a cure, sometimes it's better to just level it out and deal with the beast rather than modulate the insulin production up and down.
Does the AAV vector that you use to transport genes into the genome, does it intersect indirectly into chromosomes in a random manner?
Oh, that's a good question.
And if so, couldn't that be a danger of cancer?
That is a really good question. So he asked if AAV interacts with the genome, if it inserts genes randomly in the genome, because historically in gene therapy, that's called what's called integrational mutagenesis, and it causes cancer. There's two really good reasons that we use AAV. Number one, 80% of the population has already seen it. Almost all of you would test positive for the antibodies to have seen this virus, but you probably had no symptoms when you got it. The second reason is that if it integrates, we use a non-integrating form of AAV. We create what's called an episome, so we're not trying to integrate into the chromosomes, but when it does, it integrates into a safe site in chromosome 19, so it does not cause those issues. And lentivirus in the past, the retroviruses did cause those issues, and that was, you know, back in the 90s, and that's a pretty serious issue. So no, it does not, it has not caused cancer yet in a human.
Do you know if any of these treatments can be used to cure glaucoma or degeneration of the optic nerve?
I have a keen interest in eyes. And we just had one of our first lit reviews with medical doctors about actually macular degeneration, wet and dry. So we are looking at, we would like to try them and see if some of the regenerative gene therapies would help in some of those areas. Right now, in macular degeneration anyway, they use something called VEGF. But VEGF, what it's really doing is limiting the proliferation of veins. It's limiting vascularization. And that probably isn't where we want to go with this technology, so we do hope to try some of the regenerative gene therapies in eyes. And I have a particular interest, and this is going to sound really odd, in pentachromacy. I would like to see in billions of colors and use gene therapy as a mode and method to do that. So some creatures we see in millions of colors, some creatures see in billions of colors. And how is this applicable? In 2016, humans in a clinical trial were injected with the light-sensing algae, the genes from light-sensing algae to help them see. So there are a myriad of places that we might go and get genes to help us sort of enhance our life with a medical doctor, so you can talk about your health concerns, and then they will tell you what they suggest for the health concerns, if they suggest anything at all, depending on your medical records. So they need to work with your doctor and get medical records and find out what's right for you. And that actually plays to your question, what does it cost to do gene therapy? It's generally based on body weight, so it can vary from person to person. But they will talk to you and answer all of your questions. You'll talk to a medical doctor who gives gene therapies, and they would examine, you know, what are you trying to treat? Yeah, it's integrated-health-systems.com. It's got a bunch of dashes in it. We do have a doctor that works out of Florida sometimes, and we're building our network of doctors who will do your pre and post data. So there are a couple clinics right here in Florida where you can go to get your pre and post data, but they don't give the gene therapies. You would travel out of the country with one of our US doctors to do the gene therapies there.
I have a question from Dr. Paul.
Hi, you partially answered it just a minute ago, but let's say that I have a patient that I would like to refer for gene therapy. Is that possible today?
Yes.
What would be the mechanism to do that?
You would want to contact BioViva, so you would want to contact info@bioviva-science.com. You can email us, and then we would send the information of how you get involved with us and what the exchange of data looks like. It's a long-term commitment. You'll be dealing with the doctors that work offshore while they give the gene therapy. They'll be communicating with you what they have given the patients. They need to get all the pre-data. So a patient just can't come in tomorrow and take a gene therapy. If you're treating dementia, or you're treating muscle loss and things like that, we need images, we need blood work, we need several testing kits done before you can participate. They would do that all through you. Then they would come to us for the gene therapy, then they would come back to you, and then our contract just essentially says that for a minimum of 5 years, you will continue to follow up with these patients, do all this blood work analysis and the images and everything again, so that we can track them and see how they're performing, and that goes back to BioViva.
Could you give us a minimum or maximum of the cost? I know the weight is variable. What's the ballpark?
Oh, I could just say it goes from tens of thousands to hundreds of thousands. If somebody wants a triple gene therapy and they, the doctors agree to that and they want the higher doses suggested by clinical trials, it can become quickly cost prohibitive for most people. So that's something you would want to talk to integrative health systems about. But if they're just treating a kidney or something like that, it gets us back into a reasonable realm. And again, we can treat, I don't think every spot is open, but we could treat a few more patients for free with dementia. They are responsible for the pre and post work, but not for the gene therapy itself.
I have a question here from Melissa.
I had a couple of little questions. So the gene therapy is biological, not using nanotechnology or anything like that.
Right, so when you think about it, this is nature's nanotechnology. So it's not, there's nothing synthetic in it, though, if that's what you're asking. It's all natural human genes.
Okay, and if you have, say, you're using it to kind of refurbish the muscles and things of the body, kidneys and whatnot, to bring them back to life, maybe?
Well, with muscle mass, we know we can predictably, in 8 out of 10 people, increase muscle mass. And the reason that works is because the gene itself blocks something called myostatin. So everybody has myostatin in their body, and it limits your muscle growth. But as you get older, you're not developing muscle, and it's basically limiting it down. So it blocks the myostatin to increase your muscle mass. Something like telomerase is acting right on the chromosome by creating the enzyme in the nucleus.
Oh, thank you. And are you changing diet or making any lifestyle changes?
I ate 2 candy bars today. Oh, no. Because I had to work and I was at the hotel and I didn't know where to go eat. So you know what, the best thing to do is to do lifestyle changes and make sure that you eat healthy. I can't always do that because I'm traveling, so I like to consider myself the worst case outcome of gene therapy. But yes, and we want to get people really excited about that. We want to get you excited about your future because this isn't just something for my body. This isn't just something for bodies who can afford really expensive costs. This eventually is going to be available to you. And we want you to get excited about these genes and start picking and choosing your future. And along with that, we want you to optimize your diet. So if we had somebody who was grossly obese and we used PGC1 alpha and they could suddenly get some of that weight off and have better mitochondrial function, we hope that they would take advantage of that and be more active. And my guess is that they would, because bodies like to move. So yeah, consider, don't try to solve everything with gene therapy, but it's a nice, great, lazy way to do things.
Okay. Your slide showed clearly how death from infectious diseases have been reduced in the last 100 years, but it also seemed to show an increase in the deaths from heart disease and cancer. I wanted to ask you if that was so, and if so, do you attribute that to the modern diet or any other factors?
So the modern diet and being sedentary definitely drives the diseases that we die from, but I don't think that without great intervention we would die from anything else but those diseases that you saw. So the reason that they look bigger and more expansive, which you pointed out that I should have explained that, is because people are not dying from infectious disease, they're actually living to be old. So the other diseases in that graph are actually starting to take precedence over that graph. Because if you don't intervene, we know how you'll die, and it's those diseases on there. So what we're saying is the next logical step is to intervene and then reduce those diseases, and then what do we die of if you're healthy?
There's a question.
It's the next step.
Hi.
Hi.
I want to know what countries you do the gene therapy.
So most of them are done in South and Central America. And so there are several countries that people can choose from. So there are some countries that are easier for people to travel to. There are some countries that people from the US prefer, and there are some destinations that people in Europe prefer. And so we make sure that we have doctors in all of those locations to help patients.
Liz, we have time for one more question, and
Sure.
there's one here.
Hi, where are you getting the healthy genes that you're injecting and using?
Oh, that's a good question. I get asked that every once in a while. People are like, wait a minute, where'd you get that gene from? There are banks of human genes, and they're sequenced. So we have the sequence of them, and then we have a manufacturer who manufactures human-grade gene therapies for clinical trials and for drug use, actually create them for human use. And so it's a really, really expensive and process, but the genes were not taken from your neighbor's residence or anything like that. There are known human genes that have been banked. No, it's a really good question because people say, oh my God, whose genes am I getting? These are genes that you share in common with everyone in this room. They're just not upregulated in your body. Like every one of your cells has telomerase in it, but in somatic cells it's just completely turned off. So we're putting in another copy of the gene and turning it on. That's a really good question.
Ladies and gentlemen, Liz Parrish.
Oh, thank you.
So great to have you, Liz. It's been a while. I've been wanting to have you come, and so glad that you were able to be here now. I'd like to make a mention of the fact that this whole event is being video recorded, so you can watch it again on our YouTube channel. So if you'd like to watch it, go to our YouTube channel immediately after the event. It uploads. And also the PowerPoint that Liz was using is also will be able to be seen on our website, but that'll take a couple of days for us to load that up. So be sure to go ahead and take a look at that. People all over the world are watching here tonight. We've got people from Denmark, from Toronto, Ottawa, from Puerto Rico, from Colombia. All over the world people are watching, and I'm so happy to have people who are putting together groups of people making watch parties, watching the live stream, getting together, doing what we're doing here, but in faraway places. So if you have friends that you'd like to have live a longer lifetime, to be healthier, then tell them about this. Tell them about this. And also, next month, February 27th, bring a friend with you. Bring somebody that you love, that you care about, that you want to have a healthy, long, unlimited lifespan. Bring them with you next month, February 27th, on our next event. So I'd now like to bring up our next speaker, who is the founder of the Church of Perpetual Life. He's worked his entire life pioneering approaches for preventing and treating the disease of aging. He's constantly on the cutting edge of medical technologies. The founder of our church, let's welcome Bill Falloon.
Thank you, Neil. Thank you, Neil. I'll tell you, that presentation that Liz Parrish put on, and I've been sharing podiums in conferences, speaking engagements with Liz since year 2015. I mean, what you saw is so incredible. And if any of you ever doubted whether or not the mission of this church is going to be accomplished, it's going to happen. It's just absolutely going to happen. It's just a matter, are we going to live long enough to enable us to benefit from that technology? Now, Happy New Year. And I'm not saying that from a calendar change standpoint. I'm talking about a study published right around December 28th, and it got into the media right around the beginning of January. This is a study done at the Buck Institute where they were able to elongate the lifespan of the C. elegans model of aging by 500%. And in doing so, and this is the first time I've ever seen a credentialed scientist discuss this or describe it in public at all. He said the equivalency in human lifespan, if we can do this in humans, would be 400 to 500 years. Never seen that in public by a credentialed scientist or even a layperson suggesting that the lifespan potential of humans could be increased to 400 to 500 years based on what has already happened in a study in the C. elegans model. Now, what they did was introduce several different interventions, but they happen to be what you're doing already. You're inhibiting mTOR, the mechanistic target of rapamycin. And you're doing that by taking metformin. You're doing that by intermittent fasting, calorie restriction. You're doing it by boosting your AMPK to suppress your mTOR, or you're taking rapamycin directly along with calorie restriction, intermittent fasting. You're suppressing the mTOR pathway. For most people, it's too high. You're also simultaneously improving insulin signaling. insulin sensitivity. You're doing that with metformin, with intermittent fasting, calorie reduction, maybe even rapamycin. You're already doing what extended lifespan in these animal models in a huge, huge way. And what they did is simply combined the two. They combined the insulin signaling pathway enhancement through some gene alteration and the mTOR inhibition, and they were able to extend the lifespan of the C. elegans in a way that made international headline news. And this is a human translation of how we may be able to accomplish this ourselves, not to make it to 400 to 500 years, but to live a lot longer. One of our research analysts reviewed the paper, and he said, well, here's what we might be able to do. It's up here on the screen right now, and this will be available on the church website, by the way, so you don't have to read that screen and try to write down all that information. But it basically talks about intermittent fasting, which is what I've been doing for the last couple years. I typically go 14 to 18 or more hours every day without eating anything. Some black coffee, some tea, some water, and that's it. Intermittent fasting, and then I reduce my total calorie intake, and I'll probably be starting on rapamycin very soon. I'm already doing so many mTOR inhibiting type of therapies, I don't want to overdo it, and no one wants to overdo it. But between using rapamycin and intermittent fasting in a proper way, we might be able to benefit in the way that these animals did. Now, George Church in 2015 made a rather bold prediction, but after what you heard Liz Parrish just talk about, it's not so bold anymore, because he's involved in gene modification therapy in a way that he believes by year 2030 he will have a cure for biological aging, and it may even happen sooner. But to put this in perspective, this is a study published in November in the Proceedings of the National Academy of Sciences. And the objective was, could they use a single treatment, a single implementation of gene-modifying therapy, and that's what Liz was talking about, and simultaneously treat a whole lot of age-related diseases? Would this have an effect? And bear in mind, if you only reduce the risk of, let's say, heart disease, and the patient dies of cancer later on, well, you really haven't accomplished much. Well, look what they did. One single combination gene therapy, and they were able to produce these types of results. A 58% increase in cardiac function. And guess what? We get older, our heart muscle does not pump nearly as strong, as robust as it did in youth. And, well, every year in this country, 300,000 Americans die of congestive heart failure. So this one single therapy, if that were given to a heart failure patient, would save their lives. But they went way beyond that. They were able to show a 38% reduction in the marker of aortic vascular disease, and I believe that also includes aortic stenosis, very common problem with aging people. 73% in reduction in kidney atrophy. And guess what? If we're over 60 years of age, we've got a decline in our renal function. Some people have chronic kidney failure. It then progresses to a point where they need dialysis. I can't wait till those dialysis centers disappear. When we can use a gene therapy to regenerate our renal function, we don't have to worry about facing a dialysis procedure 3 times a week. And then the fourth, this is just one therapy, one gene intervention done once. It was able to completely reverse obesity and diabetes. Wow, all of that 2 months ago published, an absolutely remarkable amount of data. Now, what's so important for everyone in this room to realize, why you showed up tonight, is reaching the critical mass. Liz Parrish and I spoke at a conference in Palm Beach on December 8th called the Confidential Cures. It was put on by Dr. Al Sears, and we put on our presentations. Now, Newt Gingrich was also part of the speaker group. He was part of that group. Now, I don't know if Newt Gingrich knew anything about the prospect of human age reversal before this conference, but he sat there and listened to different speakers talk, and guess what? This is December 8th, by the way, is when we had this conference. Less than a month later, this is an influential person, bear in mind. When we start to influence people, he's in Fox News, January 5th, less than a month later, talking about the prospect of human age reversal. And this is why talking to people you know about the fact that aging is something that is controllable today with interventions like metformin and restricting our calorie intake, we can slow it down today. But when you have influential people talking about age reversal, you start to build that critical mass. Critical mass is what's going to enable this age reversal technology, be it whatever therapy works the best, to manifest in our lifetime. Michael Rose, he's considered one of the foremost experts in animal aging research. He's published numerous papers. He predicts within 100 years, physicians will be able to keep people alive indefinitely. This concept of immortality is garnering widespread media coverage. The scientists are talking about it. We are winning this war against biological aging on the front of the public relations. We're getting great public relations from credentialed people. Brand new magazine, first publishing this month on immortality, the Immortalist magazine. It's launched by Ira Pastor. He's been a long time immortalist. He emails me all the time with new findings. I email him back. So he's put together a magazine launching in January and a book on immortality. This writer went up to Silicon Valley and interviewed the billionaires who are putting lots and lots of money into age reversal research. We've spoken about these billionaires many, many times at this church. They are funding research to keep themselves and the rest of humanity from perishing from the horrific consequences of biological aging. This is all coming out in January 2020. These studies I'm talking about just occurred, were just published over the last couple months. It is absolutely remarkable what we are seeing transpire before our eyes. And you may know the Galileo program. That's like the 60 Minutes of German-speaking Europe. They were here a couple years ago filming, and they reran it the first week of January. And once again, my email box filled up from all of our European supporters saying, "Hey, Bill, we're seeing you on TV." And I responded, let them know that, well, that was actually played a year ago, but they rebroadcast that. So people in Europe got to see what we're doing at this church and what I'm doing to slow down my rate of aging and what we're advocating for science to do to make the process go a lot faster. So these are the people who I cannot imagine are in the predicament they're in with the kind of wealth that they have. They're multi-billionaires and they're dead. And they probably did very little to prevent that. Typically, billionaires go to the most high-end, conventional medical settings they can find to treat a medical problem. That's what they do. And they get good conventional medical care. Well, at some point, that care doesn't work anymore, and they die. And Liz Parrish, she's been struggling now for 5 to 6 years or longer, a long time, to raise a relatively small amount of money. Someone with this kind of money, he should have been having a team of PhDs. I mean, he can afford it. Team of PhDs investigating everything out there, and if something looks interesting, we'll just write a check. Because right now, he doesn't have any money. He's dead. Someone else is enjoying over $18 billion. And I make a big deal about this because it's almost a crime against humanity and against oneself to have the kind of money that these individuals do, and they're very generous for the most part. They're philanthropic, but they'll just write a $100 million check to a university to have their name put on a building, and the university uses it for all kinds of reasons, but they don't target their donations in a way where if you put $10 million into what Liz is doing, wow, something could happen really, really fast, because all she's doing is accelerating the research as opposed to putting a name on a new building being put up in the name of some billionaire. It's absolutely ridiculous. Now, this individual, the founder of QVC, we all don't know what that is, I think. It's on TV all the time. He was very intuitive to put that together, the whole concept. He was worth over $20 billion, and he dies at the age of 88. Of congestive heart failure. And I really doubt that he utilized a technique that we've been talking about for the last couple of years at this church, senolytic therapy, to remove the senescent cells in a damaged heart to enable progenitor cells to induce a regenerative effect. I doubt that he considered that therapy. I've been advocating it now for the last 12 to 15 months, that if someone is having a problem with heart failure, they're no longer responding to therapy, consider senolytic therapy. Heart failure chronic heart failure, it's at an epidemic level because guess what? We know people are living longer. And as they live longer, their heart muscle degenerates or they have myocardial infarctions, heart attacks. Part of their heart muscle dies. That weakens their heart muscle, so they wind up having an inability sometimes to even walk across a room without being out of breath. So it's a slow, progressive, degenerative condition that can be dealt with. It can be dealt with, and the conventional medical people have done a good job with it, along with the alternative medical people. But what happens is the therapies eventually stop working, and these people start to die. 300,000 a year. So what we need to do is simply regenerate their aging heart. Sounds easy, except there's a barrier. And that barrier is the accumulated burden of senescent cells in their heart muscle and it disables the ability of progenitor cells to restore healthy cardiac function. Senescent cells literally block the effect of cardiac progenitor cells from producing functional cells that would then rejuvenate the heart muscle. So what we're advocating, if you know people with congestive heart failure, I'll show you the protocol at the end of this presentation, consider a round of dasatinib and quercetin. Do that 2 to 3 weeks in a row and carefully measure baseline and follow up and see within a couple months if cardiac function doesn't improve. People with systolic heart failure can easily measure that through a test called ejection fraction. People with diastolic heart failure, there's other ways to measure improvements, including your ability to walk across a room without getting out of breath. But the senescent cell removal is probably the barrier that's preventing regenerative therapies from working. And senescent cells, as we know, they accumulate in our body as we age. They emit chronic inflammatory signals, and how they damage the heart and other tissues in the body is that these senescent cells, they emit protein degrading enzymes. So you've got an aged heart, it's had maybe some infarctions from coronary artery blockage or other reasons, viral infections. There's a number of reasons for a heart to fail, and you've got all these senescent cells in the heart that are precluding the ability for that heart muscle to regenerate, including emitting protein degrading enzymes that harm and damage and kill nearby cells. So we've got to get rid of those senescent cells, absolutely critical. And in doing so, we believe the progenitor cells that are still around are going to be able to regenerate the heart muscle. So a simple graphic, I've shown it before, but just to remind people, bone marrow, that's where mesenchymal stem cells, hematopoietic stem cells, that's where they originate, and they move into the tissues throughout the body, and they set up little colonies to produce progenitor cells that can then do two things. They can either self-renew into a healthy young progenitor cell, or they can differentiate into a fresh functional cell. People with heart failure, they don't have a whole lot of fresh functional cells left. The heart muscle is weakening. And as long as those senescent cells are in the way, no matter what you do, you give people stem cell therapy, you give people therapy to regenerate the stem cells, those senescent cells are going to overcome that problem, in our opinion. So this is what we suggest people consider doing in order to get those progenitor cells in the heart muscle working again. And of course, we've been talking about this for years. We do still have stem cells. Every one of us has stem cells in our bodies. The problem is they're getting old like our functional cells. So we boost AMPK. Do that by intermittent fasting. Do it by calorie restriction. Metformin, suppressed mTOR-C1. Now, rapamycin does that directly, but by boosting AMPK, you indirectly suppress mTOR. Lots of ways to replenish NAD and activate sirtuin proteins. Again, calorie restriction, resveratrol, a number of different factors that almost everyone in this congregation has been using for decades. You are helping to sustain stem cell function within your own body, whether you know it or not. So we know that senescent cells block the ability to rejuvenate aging, degenerated heart muscles. We know that, but we also know how to get rid of that. And the dasatinib and quercetin, that combination is currently the gold standard. Now we're looking at other senolytic compounds like fisetin, evaluating them, but we already know that the dasatinib and the quercetin works. But this is a study that I put up here a couple months ago, and it talks about if you're over 70 years of age, half of your cardiac progenitor cells, they're senescent. Wow, half of them are senescent. How would you expect your heart muscle to ever repair itself? It's not. It's not unless you get rid of those senescent cells. Good news came out about a year ago where they did some research in people with pulmonary fibrosis. They were able to see some good results there, but they were also able to see heart failure reversed in the animal model in response to senolytic therapy. Senolytic therapy that we're suggesting people who have heart failure and want to be a little adventurous, try some senolytic therapy, let us know how it works. The scientists believe this could be a therapy of the future, but with 300,000 people dying every year of heart failure in this country alone, that delay every day, every week, every month results in huge numbers of people needlessly dying. So these findings on senolytics are consistent. We continue to see new evidence. I just got a paper sent in today that was published a few days ago indicating how the removal of senescent cells could help with certain female reproductive disorders. Absolutely incredible what's happening in the field of medicine. We go back to the founder of QVC, worth over $20 billion. Now, why didn't he write Liz Parrish a $10 million check to accelerate her research? I don't know why, because $10 million, you understand when you have that kind of money, that's kind of what you earn every couple days in interest income. You know, I mean, it's just, and if he's still with QVC, he's making more than that. And so every year he lived, he may have accumulated another $2 to $3 billion. And this is what the billionaires, some are investing money and donating. We highlight those. But most of them still aren't doing this. And they're letting themselves needlessly die prematurely, and they're letting the rest of us, unfortunately, suffer too as we try to raise money. So the protocol, we've put this up on the board many times. It's simple. Dasatinib is a leukemia drug. It's a chemo drug, and I know that scares people, but we're talking about giving a dose of dasatinib that's a little bit higher than what a leukemia patient might take every day, just one time, week 1, and then repeat it week 2, along with a very high dose of quercetin. Dasatinib has specific effects against senescent cells. It's a very specific senolytic, but by itself it doesn't work nearly as well as opposed to putting it with quercetin, which of course is an over-the-counter dietary supplement. So the combination of the dasatinib and quercetin is the current gold standard for senolytic therapies, and it's being recognized in the conventional medical journals. JAMA, the American Medical Association, they believe senolytics may be a medical treatment. It may treat and reverse multiple chronic diseases. We've got to do that, by the way. If we cure kidney failure and a person dies of a heart attack a couple years later, we haven't accomplished much. But again, the major medical journals are recognizing that aging is a reversible process. New England Journal of Medicine, just about 4 months ago, talking about senolytics being able to potentially reverse aging in people. We're getting the kind of recognition that we've long sought. But it doesn't do this individual any good. He's worth $1.5 billion. He died of a problem, lymphoma. Now, they're curing about 70% of the non-Hodgkin's lymphomas. They cure over 80% of the people with Hodgkin's lymphoma, but nonetheless, a number of people still perish. And I don't think he understood the concept of BCL2 inhibition. BCL2 inhibitors, and BCL2 stands for B-cell lymphoma. B-cell lymphoma inhibitor. So when you take dasatinib, you are inhibiting the BCL2 protein. And in doing so, you are not only getting rid of senescent cells, as a cancer patient, you may get some specific benefits. And if these people who have a lot of money would just look at the scientific literature, they'd be telling their doctors, prescribe me some dasatinib, I'll go out and get my quercetin on my own, and I'm going to try this to see if I can't augment whatever conventional therapy you're prescribing for me. Because I think a lot of people dying from these hematological malignancies, they're doing it needlessly. They don't have to die, but they're also not looking beyond the scope of just the mainstream people. Paul Allen, founder of Microsoft, worth over $26 billion, and he dies of lymphoma. He dies, and he was around my age. In fact, he was my age. I'm 65 years of age. Can you imagine if I had anywhere near his money and I was diagnosed with lymphoma, what I would be doing? Well, and we're already, by the way, doing this with some high-end people. You hire a team of PhDs, okay? That's your filter, because there's going to be all kind of people saying, I can cure it with this and that. Most of it doesn't work. Your PhDs are your filter. You pay them a decent amount of money, and you invite people to submit their proposals for what could potentially save your life. And I doubt that any of these people did any of that. They just don't think outside the box. I have yet to see a full-page ad in the Wall Street Journal where some billionaire says, I'm going to pay a lot of money for anyone who can extend my life just 1 year. I'll pay you $5–10 million if you have a therapy, and then you run that approach through your team of PhDs, and maybe 100, 200 people are going to say, do this, do that, and 2 of those have potential. And you do both of them, and wow, you live an extra couple years. So you start running that ad again, saying, let's get some more ideas in here. I've never seen that happen. You can believe when I get some problem, you're going to see a lot of ads out there, because I'll be looking for a lot of creative ideas and run it by my team of PhDs and MDs to find out what may really work. So we've got an individual who died recently with a lot of money from a condition that I'm not even sure he pursued all of the alternative approaches. So a lot of people are using coenzyme Q10 to slow the progression of heart failure. It can reverse it to some degree, but at some point it doesn't continue working. Unfortunately, those senescent cells are building up, and no matter what you do, the good medications that are out there for heart failure, a lot of good nutrients, the hormone replacement, all that stuff, it works for a while, but eventually heart just gives out. Heart just gives out. So I'll conclude the service with just a long quote, and if only more people thought this way, we would accelerate our rate of aging. Leigh von Hoek, the father of microbiology. I mean, he did so much research for no other purpose than to satisfy his curiosity and then disseminate that information to other scientists who eagerly look forward to getting his drawings of how things looked under a microscope. It was an incredible amount of work he did, but that led then to the fact that we have these microorganisms that are actually pathogens in many cases, and then antibiotics were developed, and you look at how many people have been saved. Wow, hundreds of billions or more, because of an individual who's just willing to use his curiosity, his intellect, and his willingness to disseminate his findings to other people, as I'm doing on a stage, as Liz Parrish just did on a stage with that fantastic presentation. So I'm open, by the way, to any questions. That really concludes my presentation for tonight, and I appreciate the crowd. A nice group here tonight.
Okay, question here.
You mentioned metformin. I'm not sure what that is.
Metformin is an anti-diabetic drug. It was discovered in the 1920s. It got approved in England in 1957. Got approved in Canada in 1970. Americans had to wait till 1995 to get their hands on metformin. Metformin is more than an anti-diabetic drug. It's a cancer risk-reducing drug. It is a drug that probably slows down aging. A $75 million grant was awarded just a couple months ago to people at the Albert Einstein School of Medicine to do a huge study to see if metformin will not slow down and or reverse biological aging. So it's a diabetic drug that's been repurposed, and we suggested this back in 1995 when it came out. We told people without diabetes, why don't you take this drug anyway? Because it reduces your risk of progressing to type 2 diabetes, and it has all these other anti-aging benefits, and it costs very little. I suggest that everyone who can tolerate it, some people have some GI side effects. I suggest that everyone at this point utilize metformin as a way to slow down and potentially reverse certain aspects of biological aging. Publix, by the way, if you have a prescription, will give you a 1-month supply for free. That's how cheap the drug is. You have to pay for it. You're going to pay maybe $10 a month. So it doesn't cost much, and it does a lot of benefit.
Let me ask the questions go on the microphone so that they can hear on the internet. Paul, thanks. I'll be right there. Eileen?
These billionaires, were they aware of these treatments?
Well, we don't know how much, how far they reached out to identify them. I've spoken at many, many conferences, and my talks are on the internet, in which tens of thousands of people have watched them. I publish a magazine in which about 1 million people read. Liz Parrish has been nonstop talking about her approach to these conditions. I am afraid, and I've seen this with conventional people, they believe going to the top flight medical centers, Mayo Clinic, and Johns Hopkins, a whole lot of them, and some of them are really good. And I'll tell you something though, I tried to set up a cancer clinic year 2000 in South Florida, and I spent a lot of money on it, never opened it. I couldn't find the right oncologist. The really good ones were just too busy. But I interviewed a lot of them from Mayo Clinic, MD Anderson, and I was very underwhelmed, as they say. Very underwhelmed. And what all of them said to me, by the way, is, you got a great concept here, Bill. You're going to bring in everything that might work against cancer and try to cure it. We love that concept. We can't do it, by the way. We're conventional people. But we love your concept. And by the way, if anyone in our family ever gets cancer, they're going to your clinic. Now, we never were able to put that all together. Spent a lot of money, just didn't open it. But the problem is, when you're surrounded by high-level people, they're pushing you to the conventional realm of medicine. And if you need heart surgery, Cleveland Clinic is a spectacular place to go. But if you've got a problem in which conventional medicine says, well, there's limitations on how well I can treat this, you better look somewhere else. And here you have these billionaires, and they're asking all the conventional people, what can you do? And they're not looking outside the box.
The question here.
Yes, I have a question. What's exactly the relationship between metformin and the French lilac? Is it synthesized from that?
Yeah, that's where they found it. It's a compound in the French lilac. I try to tell this to people who say, I don't want to take a drug. I say, well, they originally extracted this from the French lilac, and then they eventually were able to synthesize it and standardize it into a drug. But it was used as an anti-diabetic herbal hundreds and hundreds of years ago.
Okay, so follow up. Thank you. Have there been any studies on using the French lilac for the same effect as the metformin?
I think there's been a few, but you don't want to do that when you have a standardized drug, because metformin is one of many, many compounds in the French lilac. You may have one strain of French lilac having a really high dose and some having very little. So whenever you can get a standardized dose of something, you always want to go with that standardized dose. So the best way of taking metformin, if you can, is about 1,000 milligrams extended release twice a day. That's about the best way of taking it. Other people do it differently, but that's the way to do it.
Question from John.
Thank you, Bill, for all you do. There was a great article on 60 Minutes a few weeks ago about sickle cell gene therapy and basically practically a cure. Is there anything in the works similar to that they're working on for a very specific disease?
Those gene therapies, this was a 60 Minutes broadcast, and they showed miraculous recovery using a very toxic therapy, but it nonetheless was curing people of sickle cell anemia. Well, that's what Liz Parrish is talking about. We're talking about modifying gene expression in a way that we don't age anymore. If we're too old, we reverse the process. We make our cells invulnerable to viruses. George Church predicts in 4 to 5 years he's going to be able to introduce a gene therapy in which people will no longer have to deal with viruses. And some people may think, oh, common cold isn't that big of a thing. Cytomegalovirus. Everyone heard of that in this church? Cytomegalovirus, you have it, you die sooner. It's really that simple. Epstein-Barr virus. You have it, your risk of lymphoma goes up quite a bit. There's just a lot of nasty viruses out there that we just harbor. And if we can modify our gene expression in a way that we don't get any viruses at all, we're going to add some years, some healthy years to our lifespan.
I have a question I need to ask you from the internet. It's as to where someone can get dasatinib and quercetin. Where can they obtain it?
Well, the Age Reversal, it's age-reversal.net. That is a private association. The people have a forum where they talk about all kind of different sources of dasatinib and other medications that they can obtain at low cost, and in many cases individual members will take the drug and have it analyzed to make sure that the right amount of the medication is in that drug. So age-reversal.net, the forum, there's a lot of discussions about that. There are pharmacies around the world you can order it from, and we talk about that in that forum.
And now from Dr. Paul.
Just a little clarification for the local people here, Publix and Metformin. So there's two preparations of Metformin. There's the standard short-acting, and there's the long-acting called Metformin XR. The Metformin short-acting is available free at Publix. You can get 2 a day, 60 a month, free indefinitely. That's it.
And it's fantastic. We've been talking, my different organizations, since 1995 about metformin being an effective anti-aging drug. 25 years later, a study is going to start at the Albert Einstein School of Medicine. They're going to validate whether it really works or not. This is spectacular, but that 25-year lag, plus the 37-year lag between metformin being approved in England and the United States, this is what we have to fight about. We don't have time for these kind of bureaucratic delays. We need to move the technology forward. Thank goodness we have lots of people self-experimenting and reporting back to us on what kind of results they're getting.
All right. Ladies and gentlemen, Bill Falloon. I'd like to point out again that his PowerPoint and Liz Parrish's PowerPoint from tonight will be available from our website. Everything has been recorded. If you'd like to watch it again, you can. We're going to be back here again on February the 27th. We've got a great presentation then. Be sure to bring a friend that night. And I'd like to now invite you all to join with, and you can meet with Liz and Bill downstairs where we have a reception in Liz's honor. And thank you for coming, and so great to see you.